So I will, let's go on to the plenary session with a keynote talk given by Professor Stephen Jansen. Professor Jansen is a professor of plant sciences at Ulm University in Germany. He received his master's and PhD degrees in biology from the Catholic University of Leuven in Belgium and worked as plant anatomist at the Royal Botanic Gardens in Kew in the United Kingdom. He took up a junior professorship at Ulm University in 2009, where he became full professor in 2014. Over the last 10 years, he coordinated an international team working on functional traits related to plant water transport. His works combines many aspects like field work, lab experiments, microscopy, and modeling. He was listed as highly cited researcher in 2018 and 2019 and received different awards and grants. His main research interests are comparative wood anatomy, functional plant morphology, especially with respect to plant-water relations, drought-induced tree mortality, climate change, forest decline, aluminum accumulation and heavy metal uptake in plants, which is important for everybody, tropical plant diversity, ecology, conservation, and sustainability of natural resources. So all these are very interesting subjects, important for all of us. But for the moment, today, he will tell us how plant transports water under negative pressure. Professor Jansen.
Thank you, Azita, for introducing me, and good morning, everyone. Let me upload my presentation. Okay. Okay, I'm ready. So good morning, and thank you for inviting me. I feel really honored to be here a speaker and to reach out to many of you. I wish we could have met in 3D, of course, in Edinburgh, but it is what it is now. So even this is online, I'm still excited about the whole conference. So I will talk about how do plants transport water and how do they do this really under negative pressure. It is a question, it's really a long-standing question in biology, and you would have thought, well, some plant scientists should have figured this out already 100 or 200 years ago. But we know a bit about it, but we don't know many details. I would like to start by introducing you to this tree here. This is a beech tree standing in our botanical garden in Ulm. And those of you who know where Ulm is, you might be spotting that the tallest church tower we have in Ulm behind the tree here. And this is a special tree to us, and for that reason, we have also given the tree a name, Bertie. And the tree has been equipped with some, it's not just a name, but also a face. As you can see, the students wanted to feel a bit, humanize the tree more and feel more connected to it. We have a whole bunch of sensors here. We have a set flow sensor, we have a dendrometer measuring changes in the diameter, and we have sensors in the soil. We have also an infrared camera capturing or monitoring the temperature of the leaves, which would be really nice on a summer day to visualize the cooling effect also of the tree. We are monitoring and collecting a lot of data, which is, of course, for a whole bunch of scientific questions interesting. We want to see how this particular beech tree is responding to changes in climate, how this tree will respond to the next drought periods we will have in summer. But we also want to reach out to a really broad audience, to school kids and to not just people in Ulm, but hopefully many more, by using an online platform. And you can all watch and see what Bertie is doing, how Bertie is growing, how he is responding, and we try to translate in a very easy language to even kids and non-biologists what it means to be a tree and what trees are doing, because trees are not just standing there and not doing nothing at all. There's a lot of complicated processes going on, especially if you think about transport of water. So here I have some data from the last 30 days. And what's I think exciting, as you can see, suddenly on the 10th of May this year, sap started flowing in the trunk of Bertie. And this is because around that time, the leaves had fully expanded, had flushed, and transpiration had started. We can also see here the dendrometer, or that the diameter of the trunk is increasing over the last weeks. And this is because the tree is producing a new growth ring. So one more slide, a few more words about ecosystem services of what trees are bringing us. Of course, they provide a habitat for many insects, birds, mammals, and so on. They are really important for oxygen production, capturing carbon dioxide, but also thinking about water and how much water they transpire, actually bring back into the atmosphere. I think this could be made clear by looking at these two very simple drawing of a deforested area and a more intact forest. You would have much more evaporation and transpiration because of a dense vegetation, and therefore this hydrological cycle, or the plants actually can say contribute quite a bit to their own rainfall that they get. But if you have a deforested area, you will have less water going back up to the atmosphere. You will have therefore less rain, and if there will be rain, you have a higher runoff with all the problems like leaching, the erosion, and so on. You'll have hotter temperatures. So the vegetation is really an important, bringing us a buffer capacity to buffer the climate on our planet. And thinking about how much water is going back up in the atmosphere, you could we have about 3 trillion trees on our planet. If each tree would be transpiring on a daily basis 100 liters, you end up with 300 cubic kilometers of water. That's the volume on a daily basis transpired. This is 6 times the Lake Constance, which is this lake here that you can see bordering southwest Germany. You have a bit of Austria there as well, and Switzerland. And this is a really long lake. It's about 62 kilometers long and 250 meters deep. So that's just giving you an impression of this invisible river that is probably twice as big as all the other rivers together on our planet. And that's why plants and plant climate interactions have been really important for millions of years of evolution on this planet. Why do plants transpire so much water? Well, even my kids know this. We have just a very simple experiment, 2 sunflowers, same age. But we started giving the left one less water, so more drought stress, and the right one was given more water. And you see that the growth is clearly different. Plants need water to do photosynthesis. And they do they make photosynthesis more efficient by having these tiny openings in their leaves, which we call stomata. And this helps to capture via the fusion of carbon dioxide. Okay, but at the same time, a lot of the water that they take up is transpired via these stomata. So most of the water is not really needed for metabolic processes, but is transpired by these stomata. And of course, these stomata are important pores and control valves, because in the flowering plants, this is also under hormonal control, whether they open a little bit completely or they are closed. A plant, or this tree here, and this is Bertie again, is obviously a porous medium, sitting anchored or standing in another porous medium, the soil, and its crown and the leaves, the branches are exposed to the atmosphere, a free flow domain, right? I'm showing here three cross sections, section through a root, a branch or a trunk, and a leaf. And we have the transport system. The most important transport system is the xylem, which we see here in the root. This is all xylem and water conducting cells with their secondary cell walls, the primary cell walls on the dark, the really black ones here, and then tiny openings in here. And now there are primary walls sitting here that we call the pit membranes. I'll be talking about these, the pores in here as well. And so these are the water conducting. Here's a complete opening through not just the secondary, but also the primary wall. So this is a complete perforation. We have also a little bit of xylem in this tiny vein in the leaf, but then you can see that the stoma is here, this opening, and the water pathway, water, liquid water has to be transported apoplastically, symplastically, and somewhere here near this substomatal cavity, you will have a phase change from liquid to gas phase. These short non-xylem, so if you look at the entire pathway, 90% is via xylem. Xylem in the roots, xylem in the branches and the trunk, and then in the veins. But there's 1% of this pathway, which is really short part of it, is this non-xylem tissue. And that's a really important part of it, because there we have also control by living cells, control, for instance, in the roots by lignification or suberin to block off this vascular bundle in the center, so that apoplastic transport is blocked, and you can only have symplastic transport across membranes. In these membranes, you can have water channels, the aquaporins, that can open or close, controlling water uptake. And so we also have a short non-xylem section here, or pathway in the leaves. In the water conducting cells, they have, just to give you an idea about how variable these dimensions are, they can have a diameter kind of like 10 micrometer up to 500 micrometer. They can be really short, like less than a millimeter, but over 1 meter. The point is that you never have a completely open water conducting structure or a pipe from the roots all the way into the leaf. And then if we talk about the pores in the secondary walls, these are typically less than 2 nanometers, but in these pit membranes, and I'll come back to these, they are smaller than 50 nanometers. So it's a rather complicated pathway with many different dimensions and pore scales and sizes. How do plants transport water? It's actually the sun delivering the energy, and the transpiration at the leaf level is really the driving force. Water molecules transpiring here will be kind of pulling up the liquid column, and you can see, you should imagine this as a continuous column in this non-xylem part in the leaf, through the whole xylem along. Although you have many different, many cell walls to cross and different organs, it's all interconnected to even then the water molecules in the non-xylem part in the roots, and these are the root hairs and the soil. It's transported under negative pressure. And when I say negative pressure, this is absolute values of absolute negative pressure. You can quantify it as a water potential, which is the potential energy of water per unit volume relative to pure water under reference conditions. So, and this is, the units are then joule per mole or Newton per square meter, which gives you force per surface area, which is pressure. In the soil, depending on how much water is available and soil properties and so on, for instance, the water potential could be -0.2, -0.3 megapascal, and it's typically more negative in the stem and even more negative in the leaves. So it could be -1 megapascal, and outside in the atmosphere, depending on humidity, you can have -10, -100 megapascal. So there is a water potential gradient driving the uptake and the movement, typically from the roots to the leaves, but there could also be foliar water uptake, and then you can have a reverse flow in the opposite direction. I will not be talking about that, and we also do not know too much about that. There is also quite some resistance of the water flow across, because you have to cross cell wall borders and different porous media. The more resistance you get along the whole pathway, the steeper will be, the higher will be this water potential gradient. Okay, so this sounds all fairly okay, right? But what is then now the problem? The problem is, how can the system be open to water and at the same time it's also open to gas? And why, if you have really negative, absolute negative pressure there, why is there not gas coming in and our bubbles not being formed all the time? And the bubbles would be blocking the pathway, would be interrupting the transport, and then the leaves can't do their photosynthesis and the plants can't grow. So we have been, plant hydraulics people have invested a lot of their time, I guess, over the last 10, 20 years in trying to quantify and measure how resistant is the system to failure, to hydraulic failure. How resistant is this, is the xylem to embolism formation, so the formation of large bubbles. And if we use various methods in which we manipulate the plant, we typically cut in stems. While we open it up, we introduce bubbles, we very often get artifacts, and there's been quite a bit of controversy about this. Here we have an approach, and this is the Tomcat beamline at the Swiss Light Source. So if you use micro CT, you can, in a non-invasive way, scan and visualize what's going on inside without any cutting, without any potential artifacts. And we also know that this beamline, the scanning is fast and is not inducing artifacts. So we can see here these water conducting cells in this cross section. This is not, this is a grapevine. These are images of oak. We see the large water transporting cells of the oaks that are gray, that are all water filled, except for these two. This one is filled with gas, and this one. So these two are embolized. But this is at a water potential close to -2 megapascal. And if you have more drought stress to the same plant, reaching a water potential -3.5, you can see more black dots. So there's been more embolism kicking into and making these water transporting cells unable to transport water. But what we learned is that actually the failure is really rare outside in plants in the fields. So plants have really kind of adjusted. They build in enough safety to prevent problems. So it really works. Why does it work? And I should also tell you that this sap that is transported is typically saturated with gas or even oversaturated. So we have an oversolubility of gas. And we have surfactants. This is not pure water that the plants transport. We have surfactants in there, reducing the surface tension. So that even makes it more relevant than that we need to ask the problem, why are bubbles not coming in and why is it working? We need to look a bit more at these pores, the water conducting cells in detail. Here we have a 3D reconstruction of what we call these multicellular conducting cells, or the vessels. This is in the poplar tree, and brown, that is already in the late 50s. This is only over 2.5 millimeters of a wood sample, tiny wood samples in height. But what you can see is that these conducting cells, they are grouped together, or they separate again from each other, and they run in a solitary position. We also know, we don't see them here starting or ending, but we know that when they start and where they end, they do this grouped together. They never start or end blindly. So this grouping is really important. And where they are grouped, here we have a scanning electron microscopy images, and we see these even individual cells of this multicellular transport unit. We call this a vessel. Where they overlap with each other, we have tiny little openings in the secondary wall. And this is seen, this upper image, from the inner side of the water conducting cell, and this is from the outer side. And this is where we have this primary wall, a modified primary wall, which we call a pit membrane. But it's not a cell membrane. It's not a phospholipid membrane. It's actually a meshwork of cellulose microfibrils. And this looks very flimsy here, and it's partially peeled off. This was just accidentally, happened during the preparation of the SEM sample. Okay. We try to understand in more detail the 3D structure of these openings in the cell's wall. And so we did this photographic X-ray computed tomography. This is a negative image because we made it a negative image because then you can really much more clearly see how the structure looks like here, this central chamber. So on the left, we have a water conducting cell here, and this is actually water. And on the right, we have a water conducting cell where the water is. And then you have these openings. So what is black here is cell wall, an opening, and there is this central joint chamber where also this, and I can kind of rotate this if the video works. Yes. In the center, you can kind of see where you have that line where the pit membrane is running. Okay, that's kind of the 3D structure of that, what we call a pit border. Here we have a transmission electron microscopic picture of it again. Seeing the secondary cell wall, which is overhanging this chamber. And here we have that primary wall that's modified, and that is made of cellulose microfibrils. Now, if there is gas coming in, and you have gas in plants, a lot of gas is there. If there would be gas, if there would be an embolism on the right side, but water on the left side, then the question is, how is the gas diffusing? How is it spreading to the neighboring water conducting cell? And we know that if we visualize leaves under the microscope, and this is actually an oak leaf that we put for 15 hours under the microscope, and where you have these purple colors, we have embolism. We can distinguish based on the reflection of the light whether these veins are filled with water or filled with air. And we see that these bigger ones have already been embolized, and then this tiny small one. But many of these are still not, so they're not colored here. That means they are filled with water. After 25 more hours, you can see, oh, it starts spreading here more and more, but it's a not random pattern. So it's really from, the spreading is from one conducting cell next door to an adjacent neighboring one. And after 25 hours, you see most, even the tiny little veins have been embolized. There's still a few patches that where veins or some xylem has not embolized yet. So that's important to know. And we need to understand that process if we want to understand this hydraulic failure process. Let's look a bit more at these pit membranes. So here you have a better view with this electron microscopic image of these cellulose fibers that can kind of be grouped together. And because this membrane, pit membrane, has dried up, it's also reduced in thickness. It's shrunken, and the shrinkage is by 50% in thickness. And we have also the larger pores being formed, like this one being about 100 nanometers. But that is really an artifact, because if you would look at the fresh sample that's not shrunken, which we can have here in this transmission electron microscopic image, you see the membrane kind of reaching from there to there. We wanted to know the porous medium properties of this pit membrane. We perfused samples with colloidal gold particles, and we could see that the tiny ones were going in, but the 50 nanometers one were not going in. So the sizes are below 50 nanometers, and mean pore sizes between 10 to 20, porosity pretty high, tortuosity close to 1, and constrictivity also fairly high. We know, and we see also, if you look at different species, this is here a maple, this is olive, I hope you recognize it, Mediterranean plant. So not only thicker cell walls, but also much thicker pit membranes. This one here, and I tried to have the scaling similar. This is both 1 micrometer. So the thickness of that membrane, this structure, is related to how resistant the plant is to this embolism, the spreading of embolism, which we quantify here as P50, the water potential corresponding to 50% loss of the hydraulic conductivity. So the highly resistant species are, and this is actually olive, and maple is one of the dots down here. So the thick pit membranes provide higher embolism resistance. And we wanted to understand that if you think about pore pathways and the thickness of this mesoporous medium, then you need to really, what matters is this most narrow pore constriction. And the idea was, if you increase your thickness, chances are probably higher that this most narrow pore constriction becomes smaller, right? We did this not just for pore pathways, but for the whole pit membrane level and for the whole water conducting cells, so the whole vessel at the vessel level. And here we plotted the pit membrane thickness, and we modeled the probability of encountering a really large pore. The nice thing about this model is that it's size independent, and we also were able to quantify how many pits openings in the second wells we had in a vessel for species. And this is kind of what you see. You have really this kind of 3D wave with pit membrane thickness strongly determining this probability of having a large pore or not, with all these thicker pit membranes providing high safety, I would say, and these being the leaky ones. There could be over a rather narrow range of variation in pit membrane thickness, also an effect of how many pits you have in a vessel. But overall, this effect is less pronounced than for the pit membrane thickness. If we look at what the embolism, the pressure that we need to initiate embolism, what we can measure as good as we can measure this, really, and how we model this, then we see there is a really good correlation, but the absolute values are still, we're not there. So do not agree in terms of absolute values. To model this, we used a Young-Laplace, or kind of modified Young-Laplace equation. We introduced a kappa, a pore shape correction factor. We have here the surface tension, which could be variable. We assume that alpha, this contact angle, is 0 degrees, and r is the radius of the most narrow pore constriction within an entire pit membrane. But the reason why, in terms of absolute values, there's no agreement is probably because Young-Laplace is too simplistic. We cannot capture all the details and the mechanisms that may determine the embolism spreading. So what is really happening if you have here a pit membrane and you have water on the left with negative pressure and then atmospheric pressure with gas on the right? You have surfactants coating the interface, of course. And you could think of, oh, there seems to be a pathway here. Well, the meniscus would be pulled in there. It is likely, if you think about the roof criterion, that there is a snap-off event. Whatever happens with these, you could have bubbles stuck in there, coming out, depending on how fast this all happens. That's speculative. We don't really know, but it's likely that the bubbles would be coated with surfactants. And even if they would be expanding and the coating is no longer stabilizing that nanobubble, it might also be dissolved, but it could also be leading to embolism. And the nice thing is, although that was speculation, or largely is still speculation, we started looking and searching for these surfactant-coated nanobubbles. And this was really hard to do because we did here this freeze fracture transmission electron microscopy, and we could see nanobubbles with the surfactant coat artificially kind of broken up next to it. And we can also see with a nanosite instrument, if I can get my video working, it doesn't matter. We can see with a nanosite tiny particles, which we know are the nanobubbles, and we can measure the concentration and their sizes in xylem cell. So, but these lipids are really crucial if you think about the nanobubbles. Otherwise, that wouldn't exist. The internal Laplace pressure would be too high and would make them dissolve. So, we started looking also at lipids, and this is, I have to give credit to my colleague Jochen Schenk from California State University. We have phospholipids and we have galactolipids. They are amphiphilic, insoluble lipids that have very low concentrations in the xylem set, but are there. They're coating the inner walls with their hydrophilic heads and their hydrophobic tails with their unsaturated fatty acid chains. And they are extremely efficient in changing the surface tension. This is the concept of dynamic surface tension. That means depending on how, so we have here a bubble with, let's say, this is the equilibrium surface tension of 25 millinewton per meter. If the surfactants if the more jammed together and the bubble becomes smaller, we can have some inward or outward kind of these things formed here. But then the surface tension becomes even smaller, kind of 20 millinewton per meter. Could also become much higher, up to a maximum around 60 millinewton per meter. If the bubble expands, and if lipid micelles could be integrated here, and you can see that the surfactants are really stretched apart. So the whole thing is surface tension really changes, and how this works at the interface under negative pressure with different pressure conditions, changes in pressure, changes in temperature, and so on. This is a very complicated process. Okay, if we understand, this is my last point, if we understand really how trees transport water, we should be able to copy paste this. We should be able to mimic this in terms of bionic applications. And this would be really cool to think about cooling systems for outside or indoor places, or computer systems that need some cooling and so on, or simply transport of water or liquids from A to B without using fossil fuels, right? This is another new idea that we try to build synthetic trees. This is a very old design from Dixon. So you always have a root, you have a stem, and you have leaves. This was a very successful one, but a very small one, of course, by Wheeler and Stroop with using hydrogels. And this is with multiple supply lines, ceramic disc, where you have evaporation and water is pulled up from a beaker standing on the balance below. The challenge is really, and the problem is not the leaves, not the roots, because I can think of many porous media that can be used for these purposes, but the trunk or the stem. And that is really a scientific, a material scientific problem. We try to think and work on this topic. And so the system we have in our lab is a centrifuge. It's a modified centrifuge. We can stick in here branches or any other systems, and we have, by spinning it, we create centrifugal forces. If we exceed a certain rotation speed, we can induce negative pressure, and the negative pressure will be highest in the center. At the same time, we have both ends sticking in a cuvette with some water, and the water head here is smaller and it's higher here, so you have a flow from the left to the right. So you have flow, and you can measure, you have a certain conductivity while the sample is subject to negative pressure. This was invented by Hervé Cochard. And if we're sticking a branch here of, this is another maple branch, you get this kind of curve. Nothing happens. I mean, you have maximum hydraulic conductivity until you reach kind of minus 2.567 megapascal, and then embolism kicks in until you reach 100% of loss of your hydraulic conductivity. If you put in a root, you get a very different curve. Now, the reason is, so this means immediately the system kind of fails when you come into this negative pressure range. The reason is that in the root, the water conducting cells are much wider, but also much longer, and they actually open. They are completely open. You don't have end walls. You don't have these water conducting cells ending in here somewhere. And if you have an open system, any nanoparticle could be easily coming in direct to the center and nucleating embolism immediately. That's at least our interpretation of it. You can also put in a simple capillary there, and you will see you cannot transport water under negative pressure in these. So you need to think of building in your mesoporous media or your filters, if you can call it this way. The same was tested here in for Hazel branches. Sorry, the blue line is the normal curve for a simple branch we put in, and we had ending water conducting cells. So you can transport water under negative pressure up to -1 megapascal easily, and then the system fails. But if you flush your stem with a cellulase enzyme, and the cellulase is kind of creating larger pores in the cellulose microfibrils in your mesoporous medium, then you see, paf, as soon as you reach negative pressure, it collapses, the system fails. And after having done that experiment, we figured out somebody else had already published a similar experiment that had not given the interpretation. So it kind of shows really that these porous media play a crucial role. Of course, they add resistance to the transport pathway, but they play a crucial role in making transport under negative pressure possible. At least that's the hypothesis we are working on. I would like to summarize for you that trees are really, and plants take up a central position in the whole climate change debate and questions that we are facing. Also, water transport is important for that. Why is this working? So why do plants avoid constant failure? This is probably, I try to tell you, because of xylem saps, the xylem lipids being really important. The mesoporous pit membranes provide safety. They are safety valves, at least to some extent. And something I did not talk about, but compartmentalization or redundancy of the system is also really important. I simply didn't have time to integrate it in my talk here. But if there is failure, if something, you try to minimize this to, let's say, a few branches only or a small part of your entire system so that the entire organism is not dying. And I hope you can kind of see that we need much more work, and with many of you having some excellent expertise in porous media, modeling and simulations and so on, this is exactly what is needed, I guess, to understand many more details of how these multiphase interactions are really important for water transport under negative pressure. So thank you very much for your attention. Thank you for I'd like to mention my financial support. And that was my presentation. Thank you.
Thank you very much, Stephen. The timing is really perfect. And your talk is really interesting. Many aspects everybody else is dealing with in other applications. So we have 10 minutes for questions. Don't hesitate.
Can I have a question? It's Oleg. I cannot raise my hand.
Yes, Oleg, go ahead.
Okay. Thank you. Very nice talk. Very interesting. New areas for porous media research. I have kind of a public question. In Germany on the radio, I have listened that in the last summers, very hot summers, 30–40% of the trees or more are sick now. With the knowledge you have till now, can you explain this, or you have to add more dynamic effects to be able to explain this?
Yes, we see actually that there is worldwide a lot of tree mortality, and this has been for many years. But there's been an important question to us, to plant scientists: is the mortality increasing or not? And it is. At least we have some good evidence that there is some increasing mortality. What we see in Germany and in other places in Europe is that the crown density of trees, it is a partial dieback.
Okay, so let's go on. Thank you very much, Professor Jansen.